Hello everyone, welcome to Lab 7 Overview. This lab is titled New Cells Mitosis. As you can see in the figure, this lab focuses on the cell division of eukaryotic cells. Specifically, we're going to focus on cells of our body. Imagine it's cells of your body. The majority of the cells of your body are what we call somatic cells. And these are diploid cells. I'll explain this information in lecture and to help you out with the lab. So Thursday, the information we cover should help you out with this lab. Now, these cells that make up the majority of our body called somatic cells, they go through a type of cell division that involves a type of nuclear division called mitosis. Okay, so here I'm showing you a figure of a eukaryotic cell. Okay, this right here is the nucleus of the cell. The gray area that appears in little segments with little pores in it, that's what we call the nuclear envelope. All this cluster in here is the DNA contained within the nucleus, and then this is the nucleolus. These little guys out here, well, the halos are the little sun thing they have here. That represents a region in the cytoplasm called the centrosome. And these little things here and here represent non-membranous organelles called centrioles that are required for cell division. When cell division occurs, okay, new cells are produced. But new cells can be produced one of two ways. We're covering the first way. The second way will be covered in Lab 8 as well as lecture. This first method or type of cell division that we'll talk about also on Thursday in lecture and Tuesday, a week from today, is a type of cell division that involves mitosis. Okay. Mitosis begins with one nucleus and it ends with two identical nuclei. Okay. That's a key for mitosis. It's nuclear division. However, I said it's new cells. New cells are produced through cell division. One type of cell division involves this type of nuclear division. As far as the overall cell goes, the way we go from one cell dividing into two new cells is going to require the cell or plasma membrane here. If you look down here, it pinches inward. It'll pinch inward until it results in the complete separation of these two nuclei into separate cytoplasms of separate cells. Okay? So what you're going to be doing is you're going to be labeling pics, diagrams, and you're also going to be answering questions. Listen carefully. This is not a lab. As a matter of fact, the remaining 6 labs, not a single one of these labs is a lab you can sit down and throw answers down to. If you do that, your grades will be low. You have to study the information, watch any videos, and use any lecture information where possible as supplemental material for this lab. I promise you, your grades will not turn out well if you just think you can submit anything. It won't happen, okay? You've got to go over this in detail. Cell division. It's also known as the cell cycle, and I'll show you why in a second. So you're going to hear me say cell cycle, but one and the same, cell division and cell cycle. The purpose of cell cycle has to do with the sequence of events that occur in a cycle. They can continue over and over again. Mitosis. This lab is new cell mitosis. Lab 8 next week will be new cell meiosis. The mitosis and meiosis have to do with different ways that the nucleus of the eukaryotic cell can divide in the human body. Okay, in our body, say you suffer a cut. Okay, if you've ever had a cut, sometimes you notice there's no scar. Sometimes you notice your tissue regenerates to where you can see where you had the cut. Well, what happens is those cells that were damaged are replaced by surviving cells that divide through a type of cell division or cell cycle that includes this type of division of the nucleus, mitosis. Okay? As I said, but with cell division or the cell cycle, we don't just have one nucleus dividing into two nuclei. The overall cell needs to divide. This is where cytokinesis comes in. It's division of the entire cytoplasm. And when the entire cytoplasm divides, as is showing you here by the cell membrane pinching inward, that will separate the two new nuclei into separate cells. And as I said, this type of cell division has to do with the production of somatic cells. This is going to be important, Lab 7 and Lab 8, that you understand the differences in the type of cell divisions, okay? Because then we're going to transition into DNA labs. Okay, one of the labs is going to be a cancer gene lab. You're going to have to determine if that cancer was inherited or spontaneous. So you'll see this term somatic again. Then you're going to have another lab. It's a paternity testing lab, or technically a paternity/maternity testing lab. And again, you have to understand the two types of cell division, and you have to apply them. So the labs will be building on each other. Excuse me. Okay, some terms. Listen carefully. Not just for lab, but for lecture when we go over cell division. Whenever you hear the term chromatin, we're talking about the compound DNA. Whenever you see the term chromosome or chromosomes, we're talking about the compound DNA. And whenever you see sister chromatid or chromatids, we are talking about the compound DNA. So I'm just trying to let you know these three are terms that all refer to the same compound. The difference is the shape or the way DNA is packed. Chromatin, okay, is described as DNA that is loosely packed in a cell. It takes up the whole nucleus, okay? Sister chromatids are defined more like this. They look almost like an X, okay? Chromosomes are here. Chromosomes are defined as tightly packed DNA. If you go back to this, which are sister chromatids, I'll show you over here on this figure. Down here, the best way I can describe what sister chromatids are is it's a copy of the original chromosome that this shows you up here. Okay, I'm sorry, it's the original chromosome attached to a copy of itself. So we'll talk about how during cell division, before the nucleus ever divides, all the DNA inside of the nucleus has to be replicated or copied. Okay, and when DNA is copied, we go from chromosomes to sister chromatids, the original chromosome attached to a copy of itself. But if you look here, there are two types of eukaryotic cell division. As I said, the first type we talk about involves a type of nuclear division called mitosis. Okay, this type of cell division, which I call the cell cycle plus mitosis, it starts with a single parent cell that divides, producing two identical daughter cells. Not just identical daughter cells as far as structure, but they're genetically identical. Whatever DNA the parent cell contained, okay, and the genetic information stored in that DNA that was in the parent cell, it is passed on to both daughter cells. So it's structurally and genetically that the daughter cells are identical. So this right here, the parent cell, 2N, well, I got to change the background. 2N stands for diploid cell. And I'm going to post under lecture what we're going to do on Thursday is we're going to go over terms having to do with cell division primarily. And I'll explain then what a diploid cell is. So the term 2N refers to diploid, and in this case, diploid cell. Okay. What the diploid's means, the 2N means is that in the cells of our body, you have what we call pairs of chromosomes. When the DNA tightens up, we have pairs of identical chromosomes. I'll explain it more during lecture again. But the parent cell is a diploid cell itself. It will divide into two identical diploid daughter cells. All these cells, both the parent and the daughter, are called somatic cells. It's going to be different from what we're going to talk about for the other type of cell division. However, before the parent cell can divide, like I said before, the genetic information stored in the form of DNA must be copied. We call this DNA replication. Now, they used to call it DNA synthesis, okay? But as geneticists and scientists in general started studying this process of cell division, especially in relation to cancer, they realized synthesis wasn't specific enough. Okay, replication was exactly what was going on. In other words, DNA in the form of a chromosome, that DNA that forms a chromosome that's tightly packed together, all that DNA is replicated. In other words, an identical copy is made, and it's made through DNA synthesis, resulting in sister chromatids, the original chromosome made up of tightly packed DNA being attached to the copy of itself formed by tightly packed DNA that we call sister chromatids. So during DNA replication, chromosomes are copied, forming sister chromatids. I put this figure here. Now, the reason for calling cell division cell cycle, aka just means also known as the cell cycle, is because cell division, it doesn't matter which of the two types, cell division involves a cycle of molecular events that begins with one cell and ends with two cells. Now, what they're showing you here is just the nucleus primarily, okay? But when you look at this figure, notice cell division down here, it mentions mitosis. Over here, it mentions cytokinesis. Up here, it mentions DNA synthesis, which is technically is replication, okay? What I am showing you here, okay, is a diagram representing first the two major phases that make up this type of cell division. They're called interphase and the mitotic phase. In my day, okay, in my day, interphase was believed to only take up maybe 10% of the entire time the cell was dividing, and this other phase called the mitotic phase was believed to take up to 90% of the time the cell was dividing. Okay, but as scientists became more and more familiar with this type of cell division, they found a cell spends the majority of its time in this phase and only a small amount of time in this phase. So this type of cell division called the cell cycle plus mitosis, technically it's divided up into interphase and the mitotic phase. Interphase consists of three subphases. G1 phase, where the cell is preparing to enter the second phase. Subphase, S phase. S phase is where DNA replication occurs. Every chromosome in the nucleus of our cells is copied, forming sister chromatids. Okay? So they're showing you a chromosome entering, sister chromatid coming out. Then the cell will enter the last subphase of interphase, which is G2. During G2, the cell is preparing to enter the mitotic phase. Specifically, okay, the subphase mitosis. Notice it shows you mitosis is division of the nucleus only. Okay? And what happens during this division of the nucleus is this cell is making sure it arranges all these sister chromatids consisting of an original chromosome and an exact copy. It's arranging them to where the DNA now, or these sister chromatids, can be separated, ensuring that the nucleus of each daughter cell, okay, has the same identical copies of DNA that the parent cell had, okay, except eukaryotic cells aren't just made up of a nucleus. Eukaryotic cells are also made up of a cytoplasm. So this is where cytokinesis comes in. I say this every semester, and for the love of God, I don't know why students can't separate it. Cytokinesis and mitosis are two separate subphases that make up the mitotic phase. Mitosis is just the division of the nucleus. It's not rocket science. It's just the division of the nucleus. Cytokinesis is division of the cytoplasm. In other words, the entire cell divides, separating those two new nuclei into separate cells. Okay. So then what I show you is, this is a diagram. On the next slide, what I show you is this, all this information given to you in an outline format. Here's A, interphase. Here's B, the mitotic phase. Here are the three subphases of interphase, and when DNA replication occurs, they may say duplication. Then the mitotic phase, I show you it's divided up into two subphases, mitosis and cytokinesis. Cytokinesis down here, cell membrane pitches inward, dividing the parent cell into two genetically identical daughter cells. But if you look at mitosis, okay, mitosis is actually divided up into four subphases or smaller phases: prophase, metaphase, anaphase, and telophase. I'm going to let you read through this, okay? I'll go over this information though using these diagrams. Now, before I go into those figures, there is a video that I tell you on the first page of your lab protocol. It tells you to watch this video. There are a couple of corrections, such as when the lady says fibers, she means mitochondrial. Okay? And the mitotic spindle, it forms during prophase, not when she says it does. However, the video is still a good video. She uses a chalk drawing for the cell membrane, and then she uses a chalk drawing, okay, for the membrane of the nucleus. The membrane of the nucleus is called the nuclear envelope. So don't forget that, please, because you need to know. All right. Here, this figure is showing you interphase. Okay. Technically speaking, DNA is loosely packed in the form of chromatin, but we often say it's in the form of chromosomes, which actually doesn't start forming until afterwards. But it's easier to talk about it tightly packing before entering the mitotic phase, okay? So it starts off loose and during interphase. We'll go from G1 phase to S phase. During S phase, imagine that the DNA tightly packs in the chromosomes and then it's all replicated, resulting in sister chromatids. So that by the time G2 phase of interphase is completed, okay, we go G1, S phase, and G2. By the time G2 phase is completed, all of our DNA is in the form of sister chromatids. Notice also up here, it just says interphase. This just has to do with interphase. Now it says this has to do with mitosis, beginning with prophase. During prophase, what happens is these little organelles here, called centrioles, they say centrosomes, but they're known as centrioles, okay? I won't dock you if you say centrosome. Centrosome's a region. Centrioles is the actual name. These centrioles migrate to opposite ends of the cell, and protein fibers form called the mitotic spindle. Okay, these protein fibers called mitotic spindles are formed by proteins called microtubules. So again, these protein fibers called the mitotic spindle are formed by proteins called microtubules. Now, while this is going on, okay, the cell is organizing the sister chromatids that were produced during S phase. And then as prophase continues, notice this structure here called the nuclear envelope, the membrane of the nucleus, it's disassembled, releasing the sister chromatids so that they can be attached to the mitotic spindle that's ever growing. Now, this term right here, centromere. The centromeres are represented not only here, but they're actually represented much clearer over here. Okay, I don't know why they drew the line that way. Let me see if I can do this. So where these little black dots are on the sister chromatids, those represent a region of the sister chromatids, and you even see them on chromosomes called centromeres. Okay, it's made up of protein in that area, and it's at the centromeres that these sister chromatids will attach to this ever-growing mitotic spindle. And notice the centrioles by this time have migrated to opposite ends of the cell. Okay, that's prophase. Then the nucleus of the cell, which you can't see anymore because the membrane's been disassembled, but then the nucleus of the cell will enter metaphase. What happens during metaphase? Each sister chromatid is aligned end to end. Notice the end of this sister chromatid is next to the end of this one. The end of this sister chromatid is next to the end of this one, and so on. This results in the sister chromatids being aligned in a single plane that we call the metaphase plane. Notice the centromeres here that I'm pointing at with the finger. Each sister chromatid is made up of an original chromosome and a copy of that chromosome. Each of them, or each of these chromatids, has its own centromere. So the chromatid or chromosome on this side is attached to the mitotic spindle only on this side. The other one, let's say it's the copy of the original, it has its own centromere and it's attached on this side. Now when they're sister chromatids, we call each individual one a chromatid. Together they're sister chromatids attached to their centromeres. But notice they're attached on the mitotic spindle only on one side or the other, and the reason for this is so that this spindle can play a role in separating the original chromosome from its copy to opposite ends of the cell. So what the cell is doing is arranging these replicated chromosomes called sister chromatids, okay, arranging them in a manner that it ensures the daughter cells produced are carrying the same identical DNA with the same identical genetic information, not only between each daughter cell, but from the parent cell. In other words, ensuring the daughter cells are identical. Here, after the metaphase plate is formed, the nucleus will move into anaphase. Notice here I put mitosis and cytokinesis. I'll explain why in a second. Here in anaphase, now you can see what's happening to the nucleus, even though it's not clearly defined. What's happening during anaphase is the sister chromatids have been separated into daughter chromosomes. The only time we use the term daughter chromosomes is in anaphase, and we can also use it in telophase, okay? But when you say daughter chromosome, to let somebody know that one is in cell division or cell cycle, and two, that the original chromosomes have been separated from the copies of them at the centromere. So notice, identical pieces of chromosomes, daughter chromosomes, are being pulled this way, and identical pieces are being pulled this way, with the help of the mitotic spindles, which are connected to the centrioles. So this is why the centrioles move to opposite ends. So anaphase is the separation of sister chromatids in the daughter chromosomes. Okay, and the cell makes sure it separates them to opposite ends. It doesn't want any mix-ups. We'll talk about what can happen if there's mix-ups. Now what will happen is the cell's nucleus will enter the last phase of mitosis. Remember what I said before. Mitosis is nuclear division. Cytokinesis is not nuclear division. It is not part of mitosis. Again, it's not rocket science here. Okay? Telophase is the last phase of mitosis. What happens during telophase? The membrane of the nucleus, called the nuclear envelope, begins to reform around the chromosomes. So notice, at the end of mitosis, the last phase is telophase. We don't have one nuclear envelope forming. We have two nuclear envelopes forming that are going to result in the formation of two identical nuclei. Nuclei is plural, nucleus is singular. It's going to result in the formation of two identical nuclei. So then why the heck do they put cytokinesis here with telophase? It's because the division of the cytoplasm overlaps with the last phase of nuclear division. In other words, the cell, while these two new nuclei are forming, the membrane of the cell begins to pinch inward, separating the cytoplasm of one cell, of what was one cell, into two cells, but not only separating the cytoplasm, when the membrane pinches inward, it also separates the two new nuclei that are reforming to ensure that once the membrane pinches completely across, we end up with two identical daughter cells. Each one has its own nucleus with identical DNA and therefore identical genetic information. Okay? So cytokinesis is division of the cytoplasm. It's what's happening out here with the membrane pinching in. Telophase, as I've circled here, telophase, which I've circled here, letting you know it's mitosis, well, that's what's happening right here. That's telophase. Okay? Cytokinesis is not part of mitosis, it's what's happening over here. Now you have diagrams to label. Okay, make sure you read the question carefully. If I ask you to label a diagram based on mitosis, there may be some figures you don't label because they're not part of mitosis. Read the question and answer it, okay, based on what I'm asking you, okay? Now you're also going to have figures towards the end of the lab that ask you to label stages of cells that are from an onion root tip. What stage of mitosis are they in? Well, you can see here, this is not part of mitosis. This is interphase, okay? But it'll say, hey, if it asks you to label what stage of mitosis is the onion cell in, well, then you would list one of the four phases I gave you. Now, why am I showing you onion? Because the root is where one of two places where a plant grows. It elongates, okay? What causes, in this case, an onion root tip to grow larger? It's because it's formed by cells that are all dividing. But the cells don't have to divide at the same rate. So this cell here, it's in telophase. But this cell over here, it's only in anaphase. These two cells here, they're dividing also, but they're only in metaphase. And then these two cells here, they're going to divide, but they're just in the first phase of mitosis, prophase. The bottom line is, as these cells divide, one cell dividing into two identical daughter cells, that increases the overall number of cells in this onion root tip. Thus, it increases the length of the size of it. You're going to have to label some of the stages based on the figures I gave you, what stage of mitosis they're in. But then I'm also going to give you a few pics of what is called a whitefish blastula. Everybody, blastula is a term that refers to a stage of embryonic development. In other words, we're referring to an embryo. So what you're seeing here are multiple embryos that, if everything goes well, okay, they will develop into fish, whitefish, okay? Each of these blastulas is an embryo formed by many cells at different stages of cell division. So then what I'm going to show you next is a pic at a higher magnification where it just focuses on a few cells, okay, that make up this embryo called a blastula. And it shows you, hey, here's a cell that makes up part of an embryo called a blastula, but it's in metaphase. Here's another cell that makes up the same blastula or embryo called a blastula, but it's in anaphase. Here's a group of cells that make up that same embryo. They're all part of the same embryo called a blastula, but you can see they're showing you two cells at different stages, metaphase and telophase. Okay, so make sure you label what phase of mitosis they're in. Same as usual, answer all the questions, label any diagrams. Listen carefully. Make sure your diagrams are labeled clearly, and make sure you give yourself enough time to upload your report, because sometimes this labeling doesn't work out smoothly. Sometimes you run into problems and you need to figure it out. You need to problem solve how to ensure your labels of your figures, those pics I give you, how those labels don't move and that they show up. This last lab report I graded, several people had a table where they were supposed to draw the cells between cheek cells and onion root cells, and their figures were not showing up. The tables were pushed off to the side. I couldn't see anything. I dock you points because half the class at least did make sure I could see the figures drawn out. That's on y'all. That's y'all's job to do. Give yourself time, please, okay? I got to erase this. See last slide for video. I need to get rid of this. It's not the last slide. I'll fix it. It'll be correct on the under labs that I gave you. Okay. Last. It's just the PowerPoint document. I give you some information such as watch the video. Make sure you watch that video. You probably have to watch it twice. Okay, read the background information, some of which I covered. Label the diagram. Make sure you pay attention to what it's asking you to label. I don't need a bunch of extra stuff labeled. I'll dock you if you're labeling stuff I didn't ask for. Answer the question. That's it. Okay, down here begins the next part, which leads into this diagram. Again, it says label the diagram according to mitosis. You may or may not label every single stage of the cell here. It says just mitosis, okay? And then explain the four phases. Well, that's in the PowerPoint. That's not hard to do. Over here, cytokinesis, label the diagram, what you can of the overall as well as what's inside, and then define what cytokinesis is, okay? Don't get fancy with your definitions. I mean, especially if I give it to you, just rewrite it, or rewrite it in a manner you can understand it better, okay? But don't get too fancy. Here's the part about the onion root tip and the whitefish blastula, which is an embryo, about label the stages. Make sure you read the questions so you know what I want you to label. Last, it's just asking you questions here. I do expect you to answer these questions correctly. They're not answers you can put down right away. Cell division is not an easy process. You have to study it, but we're also covering it in lecture. So don't just think of it as an investment in your lab grade. Think of it as an investment in your lecture grade too, because your lab and lecture grades go together. So you can put a positive spin on this, or you can blow it off, and it's gonna hurt you in both. Okay. Hopefully you'll put forth the effort, and hopefully I'll do a good job. Hopefully I've done a good enough job with the lab overview, and hopefully I'll do a good enough job in lecture to where you can put this all together, okay? Now remember, if some information doesn't make sense, we start covering cell division on Thursday. I'm going to go over some terms, some background terms that are related to this, okay? And then if you have any questions at the end of lecture on Thursday, even if it's lab, we can talk about it. All right, y'all take care now. That's the end of the recording. Bye.